Hello, my name is Eli Sword, and I'm a field application scientist at GenX. And what I wanted to focus this tutorial on is the sample analysis. So with that said, I'm going to jump right into the GenX analysis interpretation interface. And so just a basic orientation. In the upper right-hand corner, you can get relevant information on the subject, clinical, as well as VCF sample. If we look at the clinical information, we can see the relative phenotype, which is Noonan syndrome. We can see the filters that are applied, the protocol that was implemented, as well as the different genetic model. Those genetic models are listed on the top. So we have the fast track, we have those that are for recessive homozygous or compound het, dominant het, mitochondrial, incidental findings. If you have a CMV file that you've imported, you'll also have that displayed, and we'll focus on that in a different tutorial. You'll also notice that we have a variety of variants listed on a per row basis in our variant table. We have annotated these variants with a variety of different annotation sources, which we'll go into in a little more detail. On the left-hand side, we have the filter logic, which allows you to rapidly identify your variants of clinical significance. This filter logic is provided by the Fast Track, so we provided some default filter logic. If you wanted to look and understand a little bit more, you can hover over that individual criteria, and it'll show you what's being applied. You will also have flexibility, so if you wanted to delete any of the filters that are applied, you have the option to do so. Furthermore, if you wanted to apply an individual field from your variant table, you can click over the filter option and automatically select your specific criteria. So let's say we wanted to look at those that are associated with a pathogenic or likely pathogenic classification. You'll be able to see that in real time in the variant table, but also displayed in your filter logic. You'll also be able to see that we have information on the phenotype. So if you wanted to edit, for example, add additional phenotypes, you have the capability to do so. We have disease frequencies, as well as the ability to implement gene panels, gene lists, and variant maps. In terms of the variant analysis, we provide you with all of the relevant public annotation sources, which are updated regularly. If you click on the location of your variant, it'll display that variant in Genome Browse, as well as a variety of annotations, which can provide relevant information. You also can hyperlink out to the UCSC Genome Browse, relatively easy. In terms of the gene level, you'll be able to see information being pulled in from OMIM. So in this case, we know that our patient is affected with Noonan syndrome. We can see it's associated with the inheritance model of autosomal dominant. Similar information is being pulled in from Decipher. And we also have the ClinVar findings of the RAF1 gene that are pathogenic or likely pathogenic. We can see we have hyperlinks out, as well as 26 missense mutations associated with this condition. We also have information being pulled in from the genomic and genetic data, as well as our ACMG sample classifier. I wanted to just focus a little bit on this because it is one of our highly valued features of the software. What this does is it auto-populates according to the available evidence, the relevant ACMG criteria. So there's 28 ACMG criteria. We can see we have a variety of criteria that are already applied. If you wanted to investigate a little further, you can look at the individual evidence that is related to that particular field. You can also hover over any of the criteria and it'll identify the associated findings. You can also choose to disagree with a particular evidence, as well as save that, which will be stored for all downstream analyses. We also have variant calling, which will pull in information from your VCF file. In the evidence section, we have our phenotype-driven algorithm, which is called Phenix. Phenix is a great utility because it will rank the genes based on their association with their particular phenotype by utilizing biomedical literature. In this case, we can see the diseases related to our particular gene. If I scroll down, we can see the Noonan syndrome association with OMIM, UniProt, and Decipher. And what it's doing is it's utilizing those relevant key search term, which in this case, it's utilizing that phenotypic term. We will also display the gene summaries for RAF1, relevant publications, as well as variants related to your particular gene. We also pull in information from ClinVar, as well as display the number of publications related to your particular condition. In this case, we have 163 publications, which are associated with Mastermind. If you have a license to this, you can go into a little more detail into this application. We also by default will display the in-house allele frequency. So if your variant is observed across your internal cohort of samples, you'll be able to see the frequency in terms of the variant genes as well as the allele frequency. We also have the effect prediction. So in this case, we're looking at a missense mutation and the severity is medium, and we're looking at the frequency across different public databases. In this case, it's actually novel. So due to the high frequency as well as the pathogenic classification associated with ACMG, this would be a causal variant that we may want to report on. To report, you can click on the relevance category. It'll give you options to select the inheritance model, the relevance, as well as the pathogenicity, and pull in any relevant information from ClinVar, OMIM, as well as your biomedical literature. In this case, we'll pull in information from our Noonan syndrome. Once the relevant information has been selected, you can click on Save. And to generate the report, you can then go into the upper right-hand corner, click on the report preview, which will bring you to the report editor. Once you have confirmed that the information is correct, you can click on the Save option, which will then give you two options to render it in an Excel or a PDF format. The PDF format is displayed as such. So we have our GenX report, which is one that we provide by default. But if you have a particular lab design that you want to have integrated or a particular brand, you can definitely perform those customizations with the GenX analysis platform. So with that said, that concludes the demonstration for the sample analysis tutorial. As mentioned, if you have any questions about what was demonstrated here or questions in general, please don't hesitate to reach out to support@genx.com. Thank you for using our software and good luck with your NGS variant interpretation.